This is distillation apparatus. Okay, we have our Diet Orange Crush in here, and we're going to heat it up. So we need to turn on the gas and get some fire going. And you might be able to hear the gas. So there's our flame. Now what's going to happen to the liquid as it heats up? It bubbles. Now, what is that process? And this is one of your vocab words for Monday, when a liquid turns into a gas. Not evaporation.
Distillation.
Boiling. Okay, so it's going to boil. That's the phase change. Evaporation is when a liquid just dissolves into the air. It's still liquid, but there's small liquid drops in the air, and so you can't see it. And that's why we have humidity. So this is going to boil, and as some of it boils, not all of it has the same boiling point. Some might boil at 90 degrees. Some might boil at 100 degrees. What do you think boils at 100 degrees? Water. Some might boil at 120 degrees. Celsius, yes. So, as this boils, the vapor will come up here. It will go into this tube here. Now I'm going to turn the water from the sink on, and the water goes in here, swirls around it, and comes out here. That will help cool and condense any vapor that's boiled out. And condensing is the process going from a vapor back to a liquid, and then the liquid drip drops out of here. So guess what's going to drip drop out of here?
Water.
Water. Pure water, which is going to leave everything else behind. Okay, so as the process has been going on today, this is the regular Diet Orange Crush. How do you notice that's a little different?
It's lighter.
Which one? This?
The right one.
This is a little lighter. So this means that the other stuff's being a little more concentrated as the water gets separated out. So that's a way to separate water out of mixtures. It's also used when you get crude oil. They heat it up, and the propane comes out first, then the gasoline comes out later, and then you're left with like the motor oil, and so that's how they separate oil as well.
Okay, does anybody know what a centrifuge is used for? Any ideas? Okay, maybe if I show you. So what we have here, guys, we have two samples. Does anybody know what they are? Milk and orange juice. Okay. Okay, we have milk and orange juice. Okay. Does anybody have any of those items on your tailgate challenge? Some people? Okay. And a centrifuge, if you look at it, I'm going to kind of list it over here for you guys to kind of look at. Okay. It has little openings in which you can put the samples that are in the test tubes. Okay, and what it does is when you turn it on, it spins really, really, really fast. If you've ever been in one of those carnival, I don't know what they're called. Is it the Gravitron? Okay, where it like throws you against the wall and they drop the floor out. They used to have one at Coast. I don't know if they still do or not. Okay, I'm not positive. It might be true. Okay, same principle is in effect here. It's spinning, and what should happen with our orange juice and our milk?
Chemicals will be different.
Diffused? They'll fuse together?
Well, let's find out. Let's find out.
It is. So this is a way for us to separate mixtures. Okay, and we're going to look at it after 20 minutes. We're going to turn it on. Okay. In a real life example, does anybody know where they use a centrifuge other than in a science lab? Yes. NASA, what does NASA use it for? I'm not saying it's wrong, I'm just curious. Oh, that's taking them used to space. I don't know if it's the same principle. They just like flips them around in like all different orientations. Okay, does anybody else know? Yes. Hospitals, what do they use it for in a hospital? Blood samples. What happens when you, like, if you take your blood sample, what happens when they spin it down? It separates. Okay, if you've never seen it, it's really interesting. You get an upper coat. Okay, it's full of like your white blood cells. Okay, and your plasma. Actually, they use it when you actually give blood. Okay, if anybody's ever given blood, which you guys are probably too young for. Okay, then you get the middle part, and then you get the bottom part. What do you think the most of your blood is?
The bottom part.
What's the bottom part? Red blood cells. Okay, so you can spit them out. Okay, so when you listen to it, you can actually hear it, and you can come over and look if you want to. You can see it spins. Okay, we're going to put it on for 20 minutes, and we'll probably come back to it when we come back inside. Okay?
So we had two different substances in here. One was the orange juice.
Ew.
If you look at it, can you see that some of it went to the bottom? Yeah, what part went to the bottom? The pulp, right. And the top is going to be more of the watery part, and there might even be another slight layer you can see density-wise that might be a little bit thicker, and obviously it's tilted a little bit because it was tilted in there. If you wanted to just save all the pulp, you could now either pour it through a filter, because you guys know about filter paper from the other lab, or you could just pour it off the edge in a process that's called decanting. And that's sometimes what they use when they make wine, because what gulp might be in the bottom of wine? Yeah, the grapes and the grape skins and the pulp of the grapes. So here's our orange juice, and you can see that it's separated pretty well. The one that may not have separated quite as well, and we'll have to pass this around as well, is the milk. However, I think if you look carefully, you'll see kind of the crusty cream at the top. Okay? And so let's pass that one around. And so take a look at this, and you'll see the cream on the top. It's just a little bit more viscous, but it is less dense than the rest of it. So let's pass those around, and you can make your observations and write those down. Remember, we put the dot with the marker on filter paper, and we stuck it in a what? Solvent. And our solvent today was?
Water.
Water. Okay, so our first one is the red, and you can see the red separated into a couple different colors, mostly just red and pink. Yeah, and if you want to pass that around, you can. Here's our green.
Orange and blue.
Did we get blue and yellow to make green? Yeah, so if we are separating two things from a mixture, is that a chemical change or a physical change? It's a physical change. If we can put these back together and still have the same stuff we started with, right? We didn't change the actual composition of any of this stuff. So blue and yellow make green. We can separate them into blue and yellow, and we're good there. Here is the black. Was our hypothesis over here correct that it was all the colors?
Yes.
Yeah, and you can see one of the nice things about the black is it also tells us which colors are the heaviest. So which color is the heaviest?
Light blue.
Yellow.
Yellow or green, then yellow, then orange, then red, then purple, then blue. So it's almost like a rainbow, but you kind of cut it here. Red, orange, yellow, green, blue, indigo, violet, right? So that was the black. It separated all of them. Here is the purple, which looks like it's made of what colors?
Purple.
Purple. So it looks like they had a purple pigment, not something that was a mixture of different pigments, right? Maybe a little bit of pink in there. I don't know. I'm colorblind, so. And then here is the blue. And is there a little bit of purple in the blue?
Yeah.
Yeah, underneath. So who would have known that? So that's how chromatography can separate things. Now in biology class, you might look at the pigments that make up the colors of the leaves. What color are most leaves on trees?
Green.
Green. Are all leaves green?
No.
What's something that has red leaves? What? Fall. A maple tree in the autumn does, right? The red maple. Roses have red leaves. And even in the autumn, you know, you can see that the leaves turn orange and yellow. Those colors must have been in there someplace. So if you actually do a leaf, you'll see not only the green, but a little bit of that orange and yellow that's in the leaves. That's hidden until autumn. And when does autumn start? Tomorrow. Okay, so that's chromatography, everybody. So to summarize, we found 5 ways to separate mixtures today. First is distillation, where you heat up the liquids and separate them by their boiling points. The water and the perfume in the Diet Orange Crush had a lower boiling point than the crush part, and that's why that boiled off first into the beaker. We can filter things. We learned about that last week using filter paper. We used a centrifuge to spin things real quick, and the heaviest stuff sinks to the bottom, the lightest to the top. And since the top is often liquids, we can decant the liquid off and pour it off if we'd like to, or you can pour it through a filter again. And the last one was chromatography, where we looked at the different rainbow colors and separated the pigments on the filter paper by using something that dissolved them, a solvent, to dissolve those.
Okay, so again, write down, guys, real quick, how we set it up. Okay, are the Mentos moving around so you can see what they look like pre-experiment? So we're going to look at a post-experiment. We know what Mentos look like.
Yeah.
I want you to be able to compare. And I'm going to say I'm going to have you guys move back further even.
because you'll get dirty.
You'll get dirty too.
Yeah, I really don't want it to get there.
It's got a long string actually.
Can I get it?
Yeah. Now this is the first effort on the Mentos geyser, so if it doesn't work, it doesn't work. Watch out, you have a bee on you right there. You guys ready?
Got me too.
Please do. Please do. For last chance.
Run, Mrs. O'Brien.
I know, and I had the wrong shoes on
Look
for this.
here.
Guys.
It's like a grenade.
I got the right shoes.
We struck oil!
I think we can do more than 8.
Mentos react with Diet Coke. So what type of change is that going to be? If it's reacting? Chemical change. Right. What is going to be the evidence of this chemical change?
Explosion.
Which of the 3 that were on the board?
The production of gas.
Production of gas.
Changes color.
Wait.
Ready?
Wait.
Go. Aim.
No.
Okay, you guys might want to back up just a little bit. Earlier today, we only did it with like 6, and this is all the way filled with Mentos. Are you guys ready?
Fire.
Ready, set, go. Oh. It blew the spout off, didn't it?
Hey, so how many of you guys have seen the Mentos people? They go on TV, they make these great things to music and all that fun stuff. Hey, we're not cool like that. Okay, but we do have, and somebody's probably making a lot of money on this, guys, a lovely little geyser tube, and we're going to hook it to the Coke Zero. We're trying different types of it. This is the Coke Zero, and we're going to see what happens to the Mentos. Everybody know what a Mentos looks like before it goes into the pop bottle? Okay, we're going to compare it to what happens to it at the end. Okay, now think about with your partner, or whoever you're standing next to, what type of reaction or what's actually happening when this occurs. Okay, talk to each other.
Chemical reaction that happens when
I don't want to miss it. This one seems generally fizzier than the other. Is everybody back far enough? Are you ready?
Yes.
Yes?
5, 4, 3, 2, 1, 0.
That might be the best one we've had today. Yeah.
Okay, guys, let's watch.
So 2 clear liquids.
Oh.
Right.
That was magic.
2 clear liquids.
Are we gonna like explode or something?
Holy cow, what's going on?
And that's the formation of a precipitate, which we'll talk about in a minute. And 2 clear liquids. Oh, well, I guess the other one's a little blue.
Oh my.
Okay, you guys get it now?
Red, white, and blue.
Yeah, red, white, and blue.
Let's go.
3 ways to tell if a chemical change has occurred. One is a color change. And remember, this has to be a novel color. It can't just be from mixing 2 different colors. So when we mixed the 2 different clear liquids to get the red, that was a color change. When we mixed the light blue and the clear and got a dark blue, that was a different color change. However, just mixing yellow and blue markers is not a color change. That's just a mixing of 2 colors. The second one is production of gas. We saw that with the Mentos and the Diet Coke, and that shot up real high. Keep in mind that's not when things are boiling. That's just a phase change. So you have to have a new substance being formed. When you boil something, it's just going from liquid to gas in a phase change, and it's still the same material. It's just in the gaseous form. And the last one is the formation of a precipitate, and that's what we did when we mixed the white color that you saw. And we'll show you what the precipitate looks like on Monday as it separates out because it's a suspension.